for joining this Benchling webinar, Powering the Fermentation Science Behind Future Foods. My name is Stella Howard, and I am a marketing associate here at Benchling, and I will be moderating today's webinar. So I am really looking forward to this discussion today that not only briefly covers Benchling's capabilities for strain engineering and fermentation sciences, but also how innovative life science companies, such as our guest Clara Foods today, are redefining the traditional food production paradigm by advancing fermentation sciences at their company. So we will get a chance to properly meet our guest speaker a little bit later today, but I am excited to briefly introduce Micah, who is a process associate to Clara Foods, which is one of the founding food technology companies in the space, and actually the first company ever to produce an animal-free egg protein on an industrial scale through fermentation. Micah has a bachelor's in biochemical engineering and has held fermentation and bioprocessing roles in a range of organizations prior to Clara Foods. Really excited to have you here today, Micah. How are you, and where are you joining us from?
Doing well. Thanks for the introduction, Stella. I'm joining you guys from Oakland, California today.
Awesome. And next, I'd like to introduce my colleague, Prem Mohanty, who is one of our senior product marketing managers here at Benchling, with over 13 years of industry experience across biopharma R&D and software product marketing. His work at Benchling focuses now on our solutions program, where he curates internal training and enablement programs for the very wide range of verticals that Benchling can be used in, such as strain engineering and fermentation, for example. Thank you so much for being here today, Prem. How are you, and where are you joining us from?
Hi, good morning, Stella and everyone else. I'm joining from Dublin, California.
Awesome. And then finally, I would love to introduce Thomas McCrory, who is a scientific solutions consultant here at Benchling, and who will be giving our live demo at the end of the webinar. Thomas has a PhD in pathobiology and over 5 years industry experience in GMP manufacturing and technical product sales. And his expertise now is at creating a variety of scientific environments on Benchling for potential clients. It's great to have you, Thomas. And how are you, and where are you at this morning?
Yeah, doing well, Stella, and welcome. Thanks. I'm joining you guys from just a bit outside Boston.
Cool. Awesome. People all over the place. Okay, Prem, do you mind taking us to our agenda? So a quick housekeeping item for today's webinar is that we really encourage audience engagement. So please feel free to use the Q&A button at the bottom of your screen to submit questions throughout the webinar. You don't have to wait for our Q&A breaks. We'll start the webinar today with Prem going over strain engineering and fermentation industry overview, followed by Micah's presentation on fermentation workflows at Clara Foods, and really some great tips for other fermentation teams out there that are looking to set themselves up for success. We'll break for Q&A for 7 minutes, and then we'll dive into the 15-minute demo with Thomas, and then we'll conclude with our last Q&A. So with that, I will hand it off to Prem.
Great. Thank you so much, Stella. Good morning, everyone. Thank you for joining us this morning. For those of you not familiar with Benchling, I'll just start off with a little bit about Benchling, and then we'll dive straight into strain engineering and fermentation. So Benchling's mission is to accelerate life science innovation for the betterment of humanity. Our company was founded back in 2012 by a team of scientists and engineers at MIT who were really frustrated with the poor quality of software they had available to support this science. So with support from top-tier investors such as Andreessen Horowitz, Thrive, and Benchmark, Benchling built out a range of solutions for life sciences on a single platform. We are fortunate to work with over 350 plus innovative life science companies, including many of the world's largest enterprises. The life science industry has evolved tremendously over the past decade. We are proud to work with several leading strain engineering and fermentation companies that have helped develop new advances. We are closely aligned with these leading innovators and work in partnership. This has helped build up our domain expertise in strain engineering and fermentation. So diving into food industry, as you all very know, food industry is massive, with an estimated $6.2 trillion in sales just in the US alone. This industry includes a diverse group of companies from food production all the way to food processing and distribution to help put food on people's plates. Food technology is a key part of this ecosystem that is helping drive innovation, especially in production and processing. Strain engineering and fermentation are at the heart of innovation in food technology. Though modification of organisms for production and fermentation are decades old practices, continued advances in the fermentation science have led to many exciting applications in foods. Here are a few of the novel foods that have been in the limelight recently, starting with cell-based meats and plant-based foods, all the way through animal-free eggs and dairy. Strain engineering and fermentation has enabled a variety of novel foods. So diving just into fermentation science, like fermentation has come a long way, starting with traditional fermentation that uses intact living organisms to modulate and process plant-derived ingredients to create food products with unique flavor, nutrition profile, or texture. For example, the use of lactic acid bacteria to produce cheese or yogurt. But we have actually moved from traditional fermentation. Now there are expanding applications across biomass fermentation and precision fermentation. So biomass fermentation, for those of you that may not be familiar, leverages a fast growth and high protein content of the microorganisms to efficiently produce large quantities of the protein. And the microbial biomass itself like serves to be the food ingredient. So an example of this would be like filamentous fungi to make meat analogs. And the example of, in case of precision fermentation, it is like, this is the one that's been like most in the limelight recently. It uses microbial hosts as cell factories for producing specific functional ingredients with improved characteristics, be that sensory or functional, such as like plant-based products or cultivated meats. So examples are like proteins such as animal-free egg proteins or cell-based meats. Based on our experience working with our customers, we see three major trends. First, we are seeing companies develop more novel strains with synthetic biology and metabolic engineering technologies. Second, bench scale technologies are getting more sophisticated with the adoption of high throughput lab automation. And finally, the data being generated, especially with scale-up studies, are becoming more complex. There is a variety of both online and offline data sources that need to be brought together to make scale-up decisions. This complexity has ushered in a new R&D paradigm. So this new R&D paradigm really requires a modern digital solution. When you think about the ideal traits of that any digital solution should have in R&D, there are really four core attributes to consider. First, it has to model the science you focus on, which means having the ability to build data models that fit your science. Second, it has to support the processes and workflows that your team performs and have the ability to scale, iterate, and evolve over time. Third, it has to empower users with data at every level, giving scientists insights about their experiments and team leaders data on their programs so they can make both scientific and operational decisions quickly with complete and accurate data. And finally, it ensures that users will immediately recognize the value of the solution, see that it saves them time, and adopt it in a way that will help them keep them compliant and consistent with the rest of the R&D organization. So the Benchling Life Sciences R&D Cloud is designed to give you a complete view of your R&D innovation. Benchling's applications span the R&D process from experimental design, in silico biology, sample tracking, all the way to process management. There are 7 applications that Benchling offers, but they are all offered from a single interface. This makes it easy for users to leverage what they need without leaving the software. All of these applications are built on a common unified platform. They share a configurable data model that maps to each team's unique scientific and operational requirements. Benchling is built on a modern cloud infrastructure, fully managed by Benchling, and offers state-of-the-art security and control. And we know that most companies have a rich ecosystem of instruments, software applications, and data stores that their R&D depends on. So we provide an open platform with full set of REST APIs and out-of-the-box connectors for lab automation, as well as ways to move data onto and out of the platform. With that, I would like to hand over to the presentation to Micah. Thank you for your attention.
So yeah, thanks guys. My name is Micah Marmerstein. I'm a process associate 2 at Clara Foods. I've been with the company for about 9 months, and I was lucky enough to join Clara right when Clara was graduating from using just the Benchling notebook to rolling out more of Benchling's applications like the registry and the request system. And I got to be really a big part of developing that infrastructure. And that's what I'm here to tell you guys about today. Tell you about how Clara uses Benchling to improve data access across the company, streamline operations. But first, I'm going to tell you guys a little bit about who Clara is and what we do. So yeah, thank you guys so much for listening this morning. And I think we're going to go through any questions now.
Thanks so much, Micah. Yeah, we have some questions pouring in for you. I'll start with the first one. This is a good question from Dan. Do you ever have to go back and add more parameters that you maybe didn't realize were variables in the past, essentially that you now want to measure? So is that possible?
Sure. Yeah. So I would say that's something that's really easy to do if you didn't change that variable very much in the past. Like, let's say you had a really stable media and you thought, this is the media that I'm going to be using forever. And so you didn't make it a part of your fermentation. Benchling app actually has this feature called a bulk edit in the registry so that you can go to a certain entity type and create a new field and then select all the entities that were ever created for that entity type and then fill in that new field that you've created. Yeah, and allow you to capture that variable for all your historical data.
How about just from your experience in fermentation science, Micah, how are the costs different between food grade and pharma grade protein production? And what are like some key highlights of the difference in the bioprocesses?
Yeah, sure. So your rate titer and your yield are basically what defines your cost metric. So for biopharma, you know, titer is king because titer isn't titer is what is the hardest to achieve, and the price at which you can sell your protein for is so high that it doesn't matter what your yield on any of your raw material inputs are because your output material is so expensive. But for industrial biotechnology, especially food, while titer is important, your rate and your yield become more important. And with the types of organisms that are used in industrial biotechnology compared to pharma, you can get those to be higher. So basically benchling, bringing it back to how that's important, benchling, making sure that you're tracking the fields relevant to your cost metrics. Like in the case of industrial biotechnology, not only just titer, but also the rate and the yield at which you're making your product is really important.
So please keep them coming in, guys. And with that, we can hand it off to Thomas.
All right, so again, my name's Thomas McCrory. I'm here as I'm a scientific solution consultant here at Benchling with you today, and I'll be walking you through a strain engineering and fermentation environment live. So what we see here at Benchling through our many implementations is really teams struggle with several key different areas. So the first being from the strain engineer's perspective, who is designing thousands of plasmids that can really be a struggle with low throughput tools, and tracking all those DNA parts that go into the strain construction along with all of the strains that are subsequently created can be a challenge. And then next, for the fermentation engineers who are dealing with both online and offline data silos, we can make data sets that are combined so to avoid use of cumbersome or difficult to analyze data sets. And lastly, for the analytical scientist, he or she is typically getting inundated with hundreds of different samples coming off fermentation runs. So really speeding up that assay turnaround is important, and it can be a struggle. So how can we help here at Benchling? So for our first point here, the strain engineer, we can embed sequence level intelligence into the strain construction process and sample tracking. And for the fermentation engineer, those, we can unify both that online and offline data to help them to optimize their fermentation production and also subsequently down process development. And lastly, the analytical scientists to be able to improve their productivity with efficient sample handoffs, kind of like what you saw with Micah with those request forms. I'll go into that a little bit live, you'll see. So here we'll look a little bit more closely at our strain engineer. So with our strain engineer, without a database to really organize and warn of duplicate sequences, it can be a challenge to know what you have and what you need to build. So if you can imagine a database that ensures metadata and sequence level uniqueness. And furthermore, it can be slow and error prone to design these plasmids for transformation manually. So we're looking to a way to design these and analyze plasmids in a more bulk fashion. And then lastly, to maintain that full sequence context for downstream processes can be difficult and error prone. So what we need is a way to really connect them and maintain that sequence level context from plasmid through strain and culture. So now I'll show you here on within the Benchling product how we've addressed these. All right. So here, quickly, this is the Benchling platform. So down the left-hand side, you see several buttons here that represent the different applications and navigation tools within Benchling. But for our strain engineer, we're going to first focus in here on the registry. So our registry or database is going to contain all of those different types of objects, which Micah had kind of alluded to in their implementation. So cultures, genes, resistances, but we're mainly interested for our purpose here is to kind of look at some plasmids. So if I search by type and I pull up all my different plasmids here, and I can also down select to specific attributes that you see are captured here in all this metadata. So we'll add here and search by a gene of interest. And in doing so, now I've restricted my search to just a single plasmid that expresses this actin-1 gene. And if we're interested in looking at this a little bit more deeply, we can click into this. So since this is an object here that has a DNA sequence, it's going to move us directly into the molecular biology tool within Benchling. And that's with that same metadata being captured and pulled forward. So these can be genes of interest or sequences that are sequence verified within our plasmid. And that really allows for scientists not to have to remember these different parts, and it provides a lineage across different plasmids. And moreover, these plasmids within Benchling are sequence validated. So you only have a single plasmid in your database that has this identical sequence, as well as your registry ID here at the top. So that really goes to solve problems for people trying to manage these with Excel or other databases where it can be quite challenging without something that's doing internal validation. So this plasmid, I generated this with our assembly wizard as kind of a one-off, but really what we need to do is to do this in a bulk fashion. So moving over to our bulk assembly tool to show you a bit about what that looks like. Here, we're going to assign a backbone and an insert from our folder structure within Benchling. So we've selected here 10 different backbones, selected our cloning methodology of choice, as well as those inserts that are going to be going into those 10 backbones. And when we go to finalize that, we see we're going to create 100 different sequences. But moreover, really, those 100 sequences are going to maintain all of that unique metadata and linkages to all those different parts within Benchling. All right, so coming back to our plasmid here, in addition to this metadata, we also have some relevant types of items that are linked to our plasmid. So if we scroll down here, we see their linkages to plasmid preps in our database, but also to some notebook entries where these things were created. So we'll go into a notebook entry here to look a little bit closer. So within a notebook entry, we see a number of different tasks, but also the experimental context in which it was created and those different parts, inserts, and backbones that were utilized. Some unstructured data capture here, as well as some more structured data capture in these different tables. So with that though, you know, for finally for our downstream team creating cultures, we want to be able to utilize that are going to be utilizing these constructs. We want to be able to maintain this this sequence and construct lineage through that culture. So to do that and surface some of that, we're going to go back to our registry here and pull up a culture that we've generated. So we'll select for our cultures here, and then further down select by adding a new metadata field for different targets. So we'll enter our same target here again and see what we surface. Okay, so we've now pulled up 3 different cultures that if we scroll over a tiny bit, you can visualize that target information. So the same actin gene that's carried forward through the constructs into the culture itself. But moreover, what makes this so useful from the scientist level is that this is all automated within Benchling. So Benchling is going to automatically detect from the sequence information, and it isn't dependent on someone needing to manually input this into Benchling. So with that, let's take a step back and look at our Fermentation Engineer. So with overwhelming diversity and large data scale data types, these can lead to errors and delays for fermentation scientists entering results. So what we really need is a way to easily standardize data capture. And typically within fermentation, experimental tracking and data collection can be disconnected. So a way to centralize online and offline data in a single source to allow for experimental changes and capture all those production steps. And lastly, to be able to optimize fermentation production, it can be complex because all of these data silos that typically exist and analyzing the trends to provide insights for finalizing your processes can be a challenge. So it's really essential for us to analyze and iterate data collection in a single place. All right, so with that, we'll look again back into the Benchling platform to see how we can address these. So going back into our notebook entry here and scrolling down a bit, we see we're going to enter into a templatized workflow for the Fermentation Engineer. So where again, we see different processing steps, but then also different registry items that have defined some culture parameters. So like Micah had shown in a very robust way, this is in a similar fashion demonstrating the culture parameters for a specific fermentation run, but also the time point samples that are collected off of the back of that run. So if we click a little bit into one of our cultures here, we can see that same rich metadata that's captured here, but also these correlated time point samples here, so that you've just seen, and these have a child relationship to that culture itself. But really, as I mentioned previously, the challenges are with correlating data for our Fermentation Engineer, and first we need to collect it. So to do so, we can do so in a structured manner right in that notebook entry. So we've got our registered cultures here, and then are able to capture some of this online data right in a structured format, capturing multiple different types of data sets, but in a structured format that then can be used to combine with other data types. These are captured within the structured results tables, but also are surfaceable right within the registry items here on these result tabs. So where all of our online data here has been captured. So we've captured some data, but really want to see the bigger picture on how what this looks like in combination with other cultures. So we can do so through the onboard analytics platform within Benchling called Insights. And if we open up our fermentation dashboard, we're gonna surface some of that information. So just as a reminder of our experimental context here, just a simple culture with a few different pHs. And if we look here at our culture, we can pretty quickly see that our culture here with pH 4 is really underperforming with low concentrations and also low OD, and that kind of correlating with our lower pH measurement for that online data. And it kind of can consolidate and centralize how we're visualizing these things, both from online data perspective, but also in tables that can pull these two things together and give us that full experimental context to make decisions on how we should be moving forward. All right, so with that, we'll look now at our analytical scientist. So our analytical scientist is, trying to address inefficient and difficult methods that are sharing these complete sequence and sample context information like location and what the samples are. So we really need the ability to share this in a complete context and not through other more disparate means. So it can also be an inefficient and incomplete view to understand the status of data generated for processes, and reporting that back up to upstream teams can be a challenge. And finally, once our processes are put in place, it can be a challenge to improve on them or see different bottlenecks down the line. So we need abilities to be able to dashboard and look at those things. So for the final time here, we'll go back into the Benchling platform, and we can look at some of those. Scroll over there. Some of those ways that we can address those in Benchling. So first, coming back here to our notebook entry to round out our last experiment with our fermentation engineer, they've now transferred their time point samples into inventory through this structured transfer table. But they want to now call out to our analytics team. So right in that structured table, we can create a request, and that's going to ask us to select our request type and pull us right into one of those forms that Micah had shown. This is obviously a more simplified version of it, but we can enter our experimental purpose here, what we're trying to have them accomplish, as well as maybe a priority level, and as long as with any other supporting information we may need. And moreover, really, we're able to do this with maintaining all of that sample context, location, sample names, and containers and container positions within a plate format. So if we were to create that, we would be able to visualize that request in our request application on the side here. So if we look a little bit into that, we see we've got a few different requests, 6 different requests that are in various stages. So in process, completed, or scheduled, as well as kind of where those samples are in that workflow. In addition to that same metadata that was captured in that form. So if we take a moment and dig in a little bit deeper into one of our requests here, that same information is then again pulled forward from our, move over to our request tab here. There you go. There's that same information pulled forward into the request itself, and with all that requester information. And our analytics scientist can now come in here and execute directly on this. So if we click the execute button, we'll see the things that are still pending, and we can create a new notebook entry to execute on this from a template. So standardized template workflow. Select our folder and create that notebook entry from a template. Now, once we come into this template, we see that, now our context for our inputs for that assay come along with us, so that we don't have to be guessing or flipping back and forth to find out what those are. And again, similar to the last notebook entry we saw, this is can be standardized tasks here, and with those structured results capture for different types of analytical data that you may capture on that. Now, lastly, one of the main challenges is, how do we report this back up? And that's done straight in a very asynchronous fashion. So if our upstream team comes into their request, they can poke at the fulfillment tab here and see all the data that was posted to this request. So these were on some other samples that were already processed, and we see some additional information that was captured here. And when this request closes out, there's multiple emails that'll get sent across. And lastly here, just to round out, we're going to look at some of the processes and how we can evaluate that through insights. So opening a process-specific dashboard, we're able to view things at a little bit of a 30,000-foot view for our group, our team as a whole. So if we can visualize how much time the strain engineer team is taking to build strains, rather, for how many fermentation runs are being performed, or requests being completed by our analytics team, so that we can assess our throughput and time to reach the team's goals, as well as looking at different individuals' contributions to those goals in a summarized table here like this. And furthermore, how these data can help to identify those process bottlenecks before they become major issues for the team. And maybe if we need to expand resources in certain areas, we could do so with some analytical information driving that. So with that, I hope you've gotten a taste for how Benchling can help the strain engineering and fermentation workflow. And we'll be able to, and I'll just kind of quickly summarize here, and then we'll break to Q&A. I definitely see a lot of questions coming in on the side here. Just so to summarize, for our strain engineer, being able to centralize DNA and parts in a centralized registry, and be able to create those things in a bulk fashion and maintain that lineage throughout your culture all the way back from your DNA. For our fermentation engineer, being able to iterate on those things faster through standardized templates and coalescing online and offline data in a single data model. And then lastly, our analytics team there, being able to have a really crisp handoff between fermentation samples and their queue that they're having to manage, and being able to identify those resource bottlenecks. All right. So with that, I'll hand that off to Stella, and we can start the last segment of the Q&A here. All right. So I can take one question here. So it's showing examples of online fermentation data. So the fermentation data, online data I showed was generally simplified. But you can structure data within Benchling in any way you'd like, right? So you can categorize it and track DO and all of those sorts of things that you might want to do in a way. In terms of how much data you want to capture, it really depends on what's meaningful for you in terms of how much online data and what interval you want to be recording that at in a structured fashion.
Awesome, thanks, Thomas. I think Micah's answered some in the thread, but I think a good one that I wanted him to kind of talk about was really, can you give examples of good decisions or correct decisions that you guys made at Clara Foods based on the metadata that you had set up within Benchling?
Yeah, I mean, I've been told to be very general about internal data, but basically what I can say is what Benchling has allowed us to do in terms of our decision making is, in fermentation we're running like back to back constantly, right? We may have a day or two in between, like when a fermentation ends and when the next one goes up. And what Benchling has really allowed us to do is, we'll have a plan for the next week's fermentation, but if we have a result that's like really interesting, we can capture all that data and do some robust analysis, like actual like statistical analysis within a day after the fermentation is done, because it's all set up and so automated that we can then, say, okay, these two experiments, actually let's scrap those and do this experiment instead because of this result that was just generated. And also having the whole database to work with instead of, just analyzing this week's runs, being able to analyze this week's runs along with the last month's runs, and do statistical analysis against the last month's run to really see if a process change made a difference or not, has been really helpful. And even when we, even when we start to like change research trajectory, like, okay, actually we're going to focus on this protein target for like the next 2 months. Benchling makes it really easy to be like, okay, now that we're done with that campaign, let's go back to what we were doing 2 months ago. Where were we at? What new insights had we generated? And let's start to iterate off that. It just makes it really easy to parse through data today and then even a year ago.
Awesome. Great answer, Micah. So with that, we will wrap up the webinar. Thank you so much, Micah, for joining us today. It was great to have you. That was an excellent presentation. I learned so much. Thank you, Thomas and Prim. And check out our upcoming events that Benchling is participating in. We're at some conferences coming up, Antibody Engineering and Therapeutics, as well as SLAS for lab automation in January, we're excited about. If you head on over to benchling.com/events, we're also running a pretty cool webinar with AWS on strategies for migrating off of legacy systems to the cloud. So with that, I hope everyone has a great day and we'll see you guys soon. Take care.